All right. Hello, everybody. Thanks for stopping by for my presentation for the Idea Exchange. My name is Joshua DiNapoli, and I am a freshman at Southern New Hampshire University. As a freshman, this is, of course, my first year attending the conference, so I'm not too sure what to expect, but I'm still very excited to see everyone's ideas, and of course, I'm very excited to present my own. Anyways, today I wanted to talk to you guys about epigenetics, what it is, why it's important, and some research I hope to pursue related to the field. All right, the first thing to know about epigenetics is that it's an incredibly diverse field. There is so much you can research and talk about. However, its variety also makes it incredibly complex. Therefore, I wanted to define epigenetics as a study and explain what molecular mechanisms are considered epigenetic. I also want to show how changes in these mechanisms can be caused by external factors and lead to disease. With all that in mind, I want to conclude this presentation by determining where my research should go from here. So let's lay some groundwork. What exactly is epigenetics? Well, simply put, the definition of epigenetics is the study of how our heritable characteristics and phenotypes, which is the sum of our physical characteristics, can be changed without actually altering our DNA sequence. DNA is, of course, a complex biomolecule that exists in every cell of our body, and it holds instructions for coding proteins that help every cell function. DNA itself is a sequence of molecules called nucleotides, for which there are 4 types: adenine, guanine, cytosine, and thymine. For the sake of this presentation, cytosine is really the only relevant nucleotide, however. Sequences of nucleotides are organized into units called genes. Genes are organized this way because one gene usually codes for one protein. While this process seems straightforward, not all genes are expressed or coded in all cells. For example, let's take a neuron, which is a specialized cell in the nervous system. While a neuron will code for proteins relevant to its cell function, it might not code for all the proteins in its DNA, say proteins that specialize for a red blood cell. Inversely, a red blood cell isn't going to code for proteins that are required for a neuron only. Epigenetic mechanisms that occur on a molecular level regulate what genes are expressed in what cells without actually changing the DNA sequence in either. Thus, epigenetics is a series of processes that all life needs to survive. However, the problem with these processes are that they are constantly subject to change by external factors, whether that be developmental, such as aging, or environmental, such as pollutants, arsenic, air pollution, even radon. This is what researchers are so concerned about. How can these factors alter epigenetic mechanisms so that they lead to disease? So at this point, some of you might be asking, what exactly are these molecular mechanisms? The answer to that is that there are several mechanisms at play. However, there are 2 processes that are incredibly fundamental to the field: DNA methylation and histone modification. DNA methylation is simply when a methyl group, or chemical group, is added to the cytosine in DNA. Methylation is important because depending on where the cytosine is in the gene, it might lead to activation, which means it codes for proteins, or oppression, which means it turns off and does not code for proteins. In other words, methylation is fundamental to regulating gene expression. While methylation is a normal process, as I mentioned earlier, it is subject to many external factors. Specifically, in the case of methylation, researchers are concerned about how certain external factors can change where methylation occurs on a gene. Hypomethylation, for example, is when a region that is normally methylated in a gene is not. Inversely, hypermethylation is when a region that isn't supposed to be methylated is. As an example of hypermethylation, let's take the promoter region of the gene, which is the starting region of the gene. This region usually lacks methylation. However, if this region is hypermethylated, it can lead to several aberrations. In fact, many scientists are starting to find out that if the promoter region of, say, tumor suppressor genes are hypermethylated, it can actually lead to cancer. And this is because the tumor suppressor genes are essentially turned off and can't stop the cancerous growth. Another important mechanism is histone modification. Histones are proteins that help package DNA to condense its length. Histones have a tail where another molecule can attach to it, usually methyl groups. When histones are methylated, they are considered modified, hence the name histone modification. The effect of the modification, whether or not the gene is activated or repressed, is a result of what type of histone is being modified, and there are several types of histones, and how much the histone is being methylated. In other words, how many methyl groups are being added to the histone. Like DNA methylation, if this mechanism is altered by external factors, it could lead to important genes being turned off in cells that need them, or it could lead to the activation of genes in cells that don't need them. Either or, that would be an issue. All right, so this is just a overall visualization of what I've been talking about so far. So here on the left, we have some environmental factors that researchers may want to study that could lead to epigenetic changes. We got diet, physical activity, chemical stress. We have the actual mechanisms of epigenetics here, DNA methylation, modification of histone tails, and then we of course have the result of this, which is changes in gene expression, and of course the changes in gene expression could lead to all sorts of diseases such as diabetes, cancer, etc. And this is the comparisons between a normal cell's epigenetics and a cancerous cell's epigenetics. So for example, for DNA methylation, as I explained in my example earlier, the promoter region of a gene is supposed to be non-methylated. However, in a cancerous cell, it is hypermethylated. Similarly, in for histones, a normal cell has histones that are modified the proper way, while a cancerous cell lacks those proper modifications overall. All right, I want to go back to my idea of connecting external factors to influencing epigenetics. And what I mean by that is I wanted to give some research linking pollutants to epigenetic change to disease. So, for example, tobacco smoke, researchers were able to sample the cells of lung cancer patients that have a history of smoking, and after looking at their blood cells, they realized that their p53 genes were hypomethylated. Now, why is that important? Well, researchers were also able to determine that hypomethylation of the p53 gene was associated with early events of lung cancer. So what you have is this direct connection between high concentration of smoke, which is these lung cancer patients, connected to hypomethylation of p53 gene, which itself has been connected to early events of lung cancer. So you are drawing that line now from the pollutant to disease. Of course, there are other examples of this as well. Radon, researchers studied patients that had high exposure to radon and noticed that there were aberrant levels of methylation on certain genes. They then were able to associate those aberrant levels of methylation on the genes to a variety of diseases. This radon is unique in these examples I brought up because the paper itself admits that its methodology is fairly flawed, the way they specifically measured radon exposure levels. But I kept it in here because I wanted to just reflect on how much more research needs to be done in this field, and how this is just simply a starting point. In fact, this is the first paper to actually study that connection. So there's still a lot more work to be done in this field, but the fact that these connections are being made is incredible enough. Finally, I want to explore this example, a black carbon. Black carbon is a particle found in air pollution. And for this research, they took the samples of about 1,100 elderly men, and they noticed that their line 1 elements, which is a region of DNA sequence, was hypomethylated. And the reason why that's important is because they were also able to associate the hypomethylation of this region of DNA to cardiovascular disease. So once again, you're connecting the high concentration of black carbon to the hypomethylation, which is the epigenetic factor, to disease. So in all these cases, epigenetics is that molecular connection between a carcinogen, disease-causing agent such as black carbon, to actual disease. And this is just explaining it on a molecular level. So that is a very, and I can't stress this enough, basic introduction to epigenetics. I really want to broaden my understanding of that field, and going forward, that is the goal of my research. And to start that, I have come up with some questions that can probably move me forward. I hope moves me forward in my research. My first question is, how can we develop therapies to actually treat these negative epigenetic alterations? And are there any available as of right now? Secondly, what other research is there on the effects of alcohol or other drugs in altering epigenetic mechanisms for the worse? As I mentioned in my last slide, tobacco smoke is a notable a drug that has been shown to cause disease, but is there more research on that matter? I would like to look more into that. Third, I am curious what further research I should be conducting in the field to learn more about epigenetics. What concepts should I be covering? What literature is really important in the history of the field and to the development of the field? And finally, I want to study related concepts of DNA damage and repair. DNA damage is happening all the time, and repair mechanisms are incredibly important to making sure that life survives. However, epigenetic changes can alter the functions of these repair mechanisms because they can actually repress the proteins necessary to create the DNA repair mechanisms. So as a result, those repair mechanisms won't be able to fix the DNA damage, and the damage will persist. So I'm very curious to studying that because I want to know what the effects of persistent DNA damage is. In fact, many researchers are trying to connect DNA damage to several diseases such as cancer. I hope to answer all these questions as I continue my research in the field, and I'm really looking forward to seeing what else it has to offer as I continue reading into it. Thank you so much for watching my presentation. I really hope you enjoyed it. If you have any further questions, please send it my way. Also, please send any feedback. You can reach me at my email, joshua.dinapoli@snhu.edu, which should also be listed where this presentation is posted. Thanks again, and have a great day.